In this video, you'll learn how to log in to your ChemiDoc, use Smart Tray technology, and acquire DNA gel images in 3 easy steps and export them. To turn on your ChemiDoc, simply push the green button in front of your instrument. When the customizable home screen appears, tap anywhere and your login menu will appear. If you're a new user, type the desired username, click the plus sign, and the new login will be entered. Administrative controls can be set by clicking the person icon in the top right-hand corner. To begin, open the imager and pull out the transilluminator. Note that all gels and blots will always be imaged on one of 3 trays. It is easy but important to clean the trays. Simply use DI water and a Chem wipe to ensure optimal performance. Smart Tray technology automatically recognizes the application-specific trays and will adjust imaging parameters and software options. For imaging cyber-safe DNA applications, use the UV tray or the optional blue tray. The blue tray protects samples from UV damage. It also allows you to visualize DNA samples without a UV shield. The UV safety shield is necessary for band excision of UV-excited stains. Center your sample on the appropriate tray. Once you are satisfied with the position, push the transilluminator back in and close the drawer. Zoom into the desired imaging area, or you can simply select Image Size. Tap Application to select the category and stain appropriate for your sample. If desired, you may take a preview of your image and set your region of interest. This can be very useful for excluding very bright areas that you won't want to quantify, such as your marker. Choose between entering your predetermined exposure time or automatic detection of your bands. Tap the camera button to capture the image. For a quick assessment of the band intensities, tap each band and the relative fluorescence units as well as percent to saturation will appear. Tap the transform icon in the lower toolbar to adjust image appearance. The adjustments will automatically be saved, but the actual data will remain unchanged. Your images will be automatically saved and exported to the gallery. Tap the image info icon. Here you can enter a custom name and notes about your image or experiment. Tap the Send Save icon to select the file format. You may export images in multiple file types. Under Export Options, tap a file type and the destination, either saving to a USB drive or to a network. When you choose the ImageLab file format, your custom name and notes, as well as all the data indicated below, are exported to Bio-Rad's license-free ImageLab analysis software, which is available for both Mac and PC. We hope that this instructional video has taught you how to capture a nucleic acid gel image. For further learning, please utilize the complete, searchable onboard manual, as well as the Teach as you Tap software assistant feature.